Thank you for joining me at the seminar today, and it's my pleasure to introduce Shannon Johnson, coming all the way from Mbari, just down the road. Shannon is a Moss Landing Marine Labs alum. Before that, she got her bachelor's in biology from UCLA, and then came up here and did a master's project on blue mussel settlement patterns. Good. She then joined Bob Reinhook's molecular ecology lab at Mbari immediately after graduation and never left. She worked there as a research technician, and she's worked extensively on population genetics and phylogenetics of benthic animals from hydrothermal vents, hydrocarbon seeps, and whale falls. She's now in the zooplankton biodiversity lab with Steve Haddock, still at Mbari, where her aim is to understand how gelatinous animals, especially ctenophores, populations are connected. And based on the topic today, it looks like there's been progress with that. Shannon brings a lively and fun approach to her science, and she has the rare distinction of having named a deep-sea hydrothermal snail in honor of a punk rock icon. That would be the, I'm going to say this wrong, but the Alvinconca strummeri, named for Joe Strummer from The Clash. Because, and I quote, "They look like punk rockers in the 70s and 80s, and they have purple blood and live in such an extreme environment." You can follow Shannon on Twitter, where she tweets under the handle Punk Rock Snails. Please join me in welcoming Shannon Johnson.
So that was like the best introduction ever. Thank you. All right, so I'm actually going to tell you guys the Alvinconca story today. So we're not just talking about ctenophores, because I need more data about ctenophores. So we're going to go back in time a little bit. So for the last 15 years, I've been studying population genetics of animals from reducing environments. So mostly hydrothermal vents, but also hydrocarbon seeps, wood, and even whale falls. And you guys have actually helped me, well, maybe your grandparents here have helped me sink some whales and study the animals that live on them. And so studying reducing environments is fantastic for studying population genetics, because they offer these like island-like habitats. And so, so this is my trophy. We get little trophies every 5 years at Mbari, and this is my 15-year trophy. And I was so stoked to see that it wasn't just the stock picture on it. And but then I realized quickly that they gave me a trophy with a bunch of projects that I haven't finished yet. So I think they're trying to tell me something. Okay. So hydrothermal vents are these really amazing systems to study population genetics on, especially in the ocean, because there are these like island habitats. And so look, oh, not even on Earth. So here's the East Pacific Rise, and it's strung from, this site is Alarcon Rise, and it's just in the mouth of the Gulf of California. We don't need all that land stuff to muddy our map, right? And then it goes all the way down, and this is the Pacific Antarctic Ridge, and it goes all the way down to off the coast of Chile. And so it's thought that many animals inhabit, and each little colored dot is a vent site that we know about. There's plenty that we don't know about. We haven't explored all of the bottom of the ocean yet, if you can believe that. But the reason why these sites are so great for a population geneticist is there are these discrete habitats, and they can be separated by large distances. They can be separated by large offsets on the ridge system, or even places like here where we have the eastern microplate. And sorry for if there's any geologists in the audience, it's this little microplate stuck between two big plates and that are submerging, touching each other. Geology words, geology words. And they actually force the little microplate up, and so it's like this bathymetrically, that word, inflated feature, and it's actually twisting. And so the combination of those things make these really strong cross-axis currents. And so the animals that inhabit vents are basically these giant tube worms, and they're essentially clams and mussels, and they are stuck to the bottom. And so in order for them to disperse and inhabit different vents, because vents don't last forever. They can last on the matter of decades in some cases. They have to disperse as larvae. And so cross-axis currents matter because then the larvae are advected off axis. And so down here, for example, the eastern microplate, this can act as anywhere from no filter to a complete stoppage of gene flow for lots of different species. And so the vents are themselves, so this is a huge chimney we found in the Alarcon Rise in 2015. And it's just smothered with these giant tube worms called Riftia pachyptala. And what's cool about Riftia is they have amazing dispersal capabilities. They are one big panmictic population from here all the way down to here. And no other animals quite do that. There's one species of Lepidodrillus limpets that maintain that same species distribution, but they do have restrictions of gene flow, of all the things we've looked at. And the Lepidodrillus limpets are these little dots on here. And I like them especially because there's actually one named after me. Nerd fame. So April already did this amazing introduction. My Twitter handle came about in a few years ago. Whenever I talk to kids, I would tell them about these snails. And so I've spent the majority of my career trying to get people excited about things like worms and snails and mussels and clams. Let me tell you, it's not that easy. And so then I started working on these Albinaconcha snails, and they're covered in spikes. They've got purple blood, and they live like in the hottest, most acidic water that any other animals except for Alvinella can exist in. And so this is the mouth of a chimney, of a hydrothermal vent chimney in the Western Pacific. And then so they make this like bullseye pattern. And so all the ecologists figured this out a long time ago. So first ring are the Albinaconcha, and then the next ring are their closest relatives, Ephemeria. And then you see little Bathymodiolus mussels out here too, and some crabs. The crabs are obviously more mobile. And the interesting thing about the Albinaconcha is we don't know a whole lot about them. They're really common, they're really abundant, but they're largely unstudied, except for that we knew that there was a lot of different species. But these species went unnamed for a really long time. We've known there are multiple species for a really long time because of molecular biology. But they were undescribed for a long, long time because their phenotype is more dependent on the geochemistry of the hydrothermal vents than it is on their phenotype. So in other words, wait, I said that backwards. Their phenotype is more dependent on, they all look the same. Depending on the vent sites, they look like that. They came from that vent site. You go to a different vent site, even in the same vent field, they look different, but they're the same species. Or they're a different species. So they're morphologically cryptic, at least to me. I have one friend who thinks that she can differentiate them, but I have proven her wrong on multiple occasions, although I would be really stoked if there were some morphological differences, because when you're out in the field, you can't just sequence them really quick for CO1 to see who you're working on. And so I'm hoping that we figure some differences out. But so far, not so much. So here's Joe Strummer from The Clash. And this was really fun to name. I couldn't believe how popular this was. I got to be on NPR and in the LA Times and the New York Times, the Washington Post. I had all these amazing people interviewing me and wanting to hear about snails. And so it was like a desperate ploy for attention for snails on the bottom of the ocean, and it worked. It doesn't always work, though, because we did just name an Ossidax species, the bone-eating worm, after Jabba the Hutt, and we got nothing. Crickets. I was like, come on, there's new Star Wars out. Don't you want to hear about the worms? No, they don't care. All right, so the Western Pacific. Remember the nice linear East Pacific rise? It's so straight. There's a couple ridge offsets. There's some interesting features. We know the geological timing of the formation of. Yeah, so the Western Pacific, not so much. All right, we're going to go back and forth through this a couple of times. So this guy, Robert Hall, is this amazing geologist, and he went through and recreated the geological history of the Western Pacific. So what you're going to see is the Pacific plate subducting underneath the Eurasia plate, and what it's doing is it's twisting all this up in a big mess. And so as we go forward in time, You can see Australia moving north, India ramming into Eurasia. Oop, too far. Okay, so here's now. It's a mess. There's all sorts of rad subduction zones, back arc basins, hydrothermal venting all over the place. It's a super fun place to work. If we take the puzzle back apart, you can see. So the reason why it started at about 55 million years ago is the lineage. I've calibrated a molecular clock for the Alvinaconchus snails, and their lineage goes back about that far. And so about 55 million years ago, they had a common ancestor. So one big panmictic population then, and becoming subdivided. Again, it's so fun. I could do this all day. I'm going to do this with networks, too. You're going to get really seasick. All right. So you can see now where the different species of Alvinaconcha are located. You can see some of their evolutionary patterns and why they exist. So there's 6 different species that we know of so far. Certainly we'll find more as we explore more places, especially in this region. There's more and more stuff. This little white dot up here is Alvinaconcha adamantis. They were just discovered a few years ago, so that's exciting. We're still finding new stuff all the time, and there's a lot more research going on up here too. Okay, so 50 million years ago, this was not so isolated. So it makes sense that these guys were reproductively isolated and diverged into a distinct species. I buy that. You guys buy that? Yeah? Okay, so also up here, this is where Alvinaconcha hessleri and Alvinaconcha adamantis live. Adamantis lives in the shallowest known hydrothermal vents in the world at 185 meters deep and goes down to about 900 meters deep. Alvinaconcha hessleri lives at about 1,000 meters deep and goes down to about 3,800 meters deep. So these guys are segregated by depth, which that's not shocking. We see species do that all the time. And also the prevailing currents isolate this region quite a bit. I believe they go like this. And so any of these larvae would be advected off here, and these guys don't even stand a chance to get back up here. And there's a really neat guy from Japan who has modeled the most amazing models. And someday I'm going to give him my population genetics data, and we're going to ground truth his models, and it's going to be such a cool paper when we finally finish this. All right, so the really interesting Alvinaconcha are the ones who co-occur. So we have Alvinaconcha bouchetti, who live in the Manus Basin, Fiji, and the Lao back arc basins. We also have Alvinaconcha kojimai, who live in the Manus Basin and the Fiji and the Lao back arc basins. And then Alvinaconcha strumeri joins them in the southern Lao basin. And sometimes, very rarely, will creep up to the northern Lao basin. And so this is my really interesting question. And I got really interested in cryptic species complexes, working on different hydrothermal vent systems and hydrocarbon seep systems. And so it's not very often in nature that we see animals that co-evolve or will undergo some sort of ecological speciation. And so I got really excited when I started seeing these guys, and that's why I started trying to recreate the history of the Western Pacific to try to understand how these things could, maybe they inhabited different vent sites and then they came back together. And so I'm just showing you guys a tiny little portion of these data because I'm the only person who likes to look at these networks. The other person's not here today. So I don't know how familiar you guys are looking at these DNA networks. So this is just mitochondrial CO1. And so each individual ball represents a unique DNA sequence for this gene. And the bigger the ball, the more individuals that had that same exact sequence. And you can almost see there's like these little hatches across. That means there's base pair differences between those balls. And the tiny balls are singletons. And so right away, so there's almost 600 sequences in this data file. So the first thing you see is there's not a ton of information in the mitochondrial DNA, which is kind of a bummer because I always want more information. But you can see that there are definitely 6 different things. And so the ones that co-occur, like bouchetti, kojimai, and strumeri, kojimai and bouchetti co-occur the most often in the most places, and they are some of the most distinct species. And then strumeri and kojimai, actually, these guys are super interesting because they're more closely related and they co-occur also pretty often. So Anything else I want to say? Any questions? Right. So the cool thing about these snails is they harbor chemosynthetic bacteria in their gill structures. And this is how they inhabit the vents. This is why they live right around the chimneys of vents. And so this is work I'm doing in collaboration with Roxy Beinart. And she published a paper a few years ago where she saw these patterns of latitudinal gradients of different geochemistry in the Lau Basin. And so she sampled a bunch of vents up here and a bunch of vents down here. She found different microbes at different geochemistries. So then she went back again in 2016 and sampled even more. And so these are the newer data. And again, we see the same thing, a latitudinal gradient of different phylotypes. And so these are 16S sequences. And so epsilon proteobacteria and gamma proteobacteria are hugely different microbes, and they do very different things. And so what's interesting is the northern Lau Basin is, okay, so these are all the sites where the animals were collected. These are the bacteria from these animals. I kind of made the colors a little too similar, sorry. Just to confuse you guys, you know, don't want to make it too straightforward. But what was interesting is, so the northern Lau Basin is dominated by epsilon proteobacteria, but with some gamma proteobacteria. And then you see a shift in the southern Lau Basin, some epsilons, but majority gammas. And then in the very southern Lau Basin, you see mostly a couple different phylotypes of gamma proteobacteria. And what's interesting is she had a predominant symbiont that she found a different gamma proteobacteria in her earlier study, and that was gone. And so this is a pretty flexible system. These animals aren't necessarily always using the same phylotype all the time. And that's an interesting thing. Okay, so now if we look at the animals in concert with bacteria, Albinaconca buscheti predominantly hosts epsilon proteobacteria. It does switch back every now and then. We got one in this thing. I have to do a bunch more sequencing. She did way more sequencing than I did. Then we have, and buscheti primarily lives in the northern Lau Basin. Albinaconca kojimai, that is kind of a little bit everywhere and also has a lot more diversity than buscheti does because it lives in more places, I think. Also has at least two kinds of symbionts and is much more flexible. And Albinaconca stremeri is less flexible and tends to stick to the gamma proteobacteria. And our stremeri is mostly limited to mostly the Tui Melilla site, but mostly the southern Lau sites. And so we've killed several million of these snails trying to figure out these patterns. And so our next step is to use a population genomics approach on both the symbionts and on the hosts so that we can not only get a really fine scale, like vent chimney by vent chimney look and see. Because my theory is, so Roxy raised the hypothesis in her last paper that the geochemistry and the microbes were actually driving the distribution of the species. And I buy that. It seems that way. Certain different bacteria do better, or I don't know. I never know what the right way to say this with the microbe people is. It used to be said that it was like the most common and abundant bacteria were the most successful. So anyways, and different geochemical conditions. So epsilons are happier in cooler, less sulfitic vents, and gammas are happier in hotter, more sulfitic vents. In general, sometimes, maybe. But I'm not a microbiologist, so I can say stuff like that and you guys can't kill me. So my theory with these guys is so the population genomics this picture doesn't quite tell me how these things diverged into distinct species, right? Like we have two really different things that live back together again. So they could have evolved in separate areas, and then there's like a secondary contact zone where they come back into contact. They're not hybridizing because they're so different. But we also have stremeri in the mix, and stremeri coexists with all of them or co-occurs with all of them, and is just is pretty flexible, or kojimai is more flexible with respect to their symbionts. So something fishy is going on, and we don't know. We need more data. and luckily we have the freezer full of it. But yeah. So this is still a huge question mark, and still has a lot of more work to do. But luckily, Corinna Brucing, who is starting her postdoc with Roxy in like 2 weeks, is going to start taking some of this stuff up too. And so she's more interested in the population genomics level, and I'm more interested, for once, in the evolutionary big picture and trying to sort out how these different things have evolved. Oh yeah, so my theory is that these guys, once they get to their vent chimney, they don't move. It's very super stable. In fact, they've gone back a year later, and they've seen the hole where they took a bunch of snails from last collection. And so, yeah, so it's like we feel a little bad about those millions of snails that we took, because we thought, oh, sorry, we're going to have an effect on that population. So I think it's this sort of level of competition. So these animals, the way they reproduce is they transfer a sperm packet. And so rather than like a lot of mollusks where they just broadcast their gametes into the water column, they are mating with their nearest neighbor. So if you don't move and you're only mating with this guy sitting right here, or girl, then you are going to be reproductively isolated because you only want to inhabit this one little vent. We'll see. We have a lot more work to do. But that's not my job anymore. This is my job. Okay, so so now I have to totally retrain my little brain how to think about population genetics. And so now I'm working on midwater systems, and it's totally different. They're not discrete habitats. We have way different barriers, potential barriers to gene flow. There are things like currents and continents. The animals I'm working on now are no longer just larvae, although they have larvae that can disperse, but now they're big and they can swim, especially when you're trying to capture them when you're blue water diving. Swim right away and then melt. And so these are my people. I wouldn't even know what a ctenophore was, or know how to get DNA out of it, or know how to sequence it without these guys. So Steve for hiring me. Yay! I kept my job at Mbari. And then Lynn Christensen, who I'm going to talk about in a few minutes, and she's our project manager and lab manager. And then, of course, Darren Schultz and Jacob. And sorry, Jacob, the one picture with your eyes closed. So anyways, none of this work would happen without all of these people. So many thanks to them. Right, so ctenophore is totally changing gears in my life and in my studies and in my little brain. So ctenophores are super cool. Like I said, I've been trying to get people excited about snails and clams and worms. Now I'm just like, look, it sparkles. Peace, I'm out. Oh yeah, it bioluminesces, it's beautiful, yeah. So now my thing is convincing people that ctenophores are also interesting. It's not just the hot chick, she's interesting too. So, all right, so this is the most recent 18S tree that's been published. And it's pretty sparse. I'll show you guys more data in a minute. And it doesn't look this bad because it's really sparse. It looks like things are kind of sorting out nicely. When you look at all the 18S sequences that Lynn has actually collected, it's a total mess, and I will show you guys that. So thanks to Lynn. Okay, that's my other story. That's not 18S, that's CON. So ctenophores are weird. They're super cool, they're super beautiful, but they're super weird. There's like the whole controversy, are they really ancient, or is their lineage really ancient? We don't say basil. Or not, who knows? They're really, really weird. They have small, compact little genomes, which is fantastic for NGS sequence, or NGS, but they're really, really freaking hard to get DNA out of. They're basically balls of highly organized, gooey water, and they sometimes have some DNA. But like for doing these next generation sequencing techniques, you need like micrograms of DNA, and I'm like super stoked when I get like 500 nanograms, like really stoked. So tricky, but they're cool. So here's CO1. You can see there's a lot of things. And CO1, Lynn has designed these amazing primers. Lynn has like fought hard to design all these amazing primers so that we can really see the diversity within the ctenophores. And yeah, it's a lot. So just let me know when you guys are done reading it. No? Okay. Okay, so here are my guys. Here's some Bolinopsis down here. There's some more Bolinopsis up here. Friends with Memiopsis. They're kind of a mess. That's okay. They're interesting. But you can see it's not just one thing over here. Whoops, that's Baroe. Somewhere around here. Oh, here we go. It's not just one thing like we see with 18S. Okay, so my new thing is also actually looking at the animals. I know I showed you guys pictures of snails. That was the only time I ever really looked at the animals. Now I really look at the animals. And that's the other hard thing, and thing that's weird about bullies and ctenophores in general is you can't preserve them. So basically you get them and you're nice to them so they don't melt. And then you try to take nice pictures of them. And this again, like thanks to the lab, because I've actually been learning how to use the camera. And so you have to like really look at them while they're still alive and take pictures of them while they're still alive, and then you freeze them in tubes. Because like you can preserve them, but then you're just going to end up with a little jar of snot. It doesn't help anybody. And so the original species descriptions, you can see these two species are really different. Like they're probably, I don't know, depending on your species concept, they're probably not even the same genera. But you can see there's not a huge number of characters to delineate different species for these guys, girls, them. So yeah, and as somebody who really doesn't do well with the morphology bits, I'm more of like a hard answer, like that's the sequence, that's the thing. It's hard, but I'm learning. And so one of the interesting things about the Bolinopsis and the ctenophores in general is that most of them are hermaphrodites and capable of selfing themselves. But we have no idea how often that happens. And I have some ideas on how to calculate that, but haven't done that yet. But regardless of if they're selfing themselves or not, they broadcast their gametes into the water column, and then they make cute little sidipid larvae. Okay, these are not actually Bolinopsis larvae. I don't have any pictures of Bolinopsis larvae. These are actually pleurobrachia, but I took the pictures and I was very proud. So, and then they have then they grow up into baby Bolinopsis. They live 1 year, which is fantastic for estimating mutation rates. I love these animals. They're so fun. But the selfing themselves is an important thing with respect to working on their genomes and looking at hybridization and looking at heterozygosity. And one interesting thing one of the students in our lab, Darren Schultz, has been working on is he's been working really hard on assembling ctenophore genomes. And he has a hard time because they're crazy heterozygous. And which he says it's not more heterozygous than most things, way more heterozygous than humans, which is what most of the software is written for, right? But that lends some evidence that they don't just self themselves all the time. Okay, and also their levels of diversity. Okay, so here's 18S. We see they're paraphyletic, it's a mess, they like switch, and they're paraphyletic with all the lobates, so not a ton of information in 18S, although it has its utilities for some stuff, right? 28S does a little bit better, delineating against different places. So this guy is collected on the east coast of the United States. These guys are all from the west coast of the United States. These guys are from Tahiti. These guys are from Hawaii. So we start to see some differentiation. And then, of course, CO1. This is just the Bolinopsis. You can see there's like a lot of different OTUs. So the two species complexes I'm going to tell you guys about today are infundibulum and vitrea. And that's because that's the most samples I have. There's lots of other things in that tree, but this is what we got. And so infundibulum is kind of the cool water species, where they've been described off the west coast of, and I lost a dot here, west coast of North America, all the way around, all up here, and then all the way to the east coast. And that's like a ridiculous species range. Nothing can do that, right? Even these guys are good swimmers. I'm telling you, they swim faster than I do. But I'm not positive that they're completely one big pedmectic population for that. The vitrea species complex, there's some in Hawaii, I can't remember where that is, the Bahamas, and then a few in Australia. Tahiti? I bet that's Tahiti. And they're the more warm water species. And these are the ones I really want to go collect. Like, don't we need to go to Tahiti, right? We're going to Hawaii in a couple weeks, so winter. Okay, so this is the network. I told you guys we love these networks. They're so fun. And this is just mitochondrial DNA. It's a little less scary to look at than that horrible tree. And so you can see there's lots of distinct things, especially think back to the Alvin and Conca networks where there was like one big ball and like 3 other balls, and there were like 200 sequences in that species, right? This is like crazy. Like, this is one collection from 1 2-week period in Hawaii. So there's a lot of diversity in these guys. This is a bunch of individuals from the Monterey Bay. Some of these guys were from Friday Harbor in the Monterey Bay. These guys go from Alaska all the way to Svalbard and Woods Hole, the east coast of the United States. Okay, so we're going to break this network up into some nicer bites, simpler bites. And so I think this is the real Bolinopsis infundibulum. It was originally described in Svalbard. I'm sure I'm totally Svalbard. Svalbard. Yep, saying that wrong. And I am lucky enough to have samples from there now, but the samples are pretty distinct. There's certainly a lot of variability within this network, but the samples go from Alaska all the way to Woods Hole and Oceanographers Canyon, which is off the east coast of the United States too. So it's not shocking that we would see this level of diversity within a thing, but it's interesting. It's really interesting. Luckily, we have collaborators up in Norway, and we are going to, they have a bunch more samples, and we have a lot of sequencing. And so we're going to fill in some holes in this network and actually get some good samples, because this is like everybody's a singleton in this network, and that sucks. Nobody wants one sample. I have it a lot. Okay, so the vitreous species complex has kind of the same story as the real Boles infundibulum. So this guy's actually a named species, Boleashlii from Australia. But you can see there's another guy from Australia, another singleton, and then somebody from the Bahamas. This guy's from Moorea, and these guys are from Hawaii. So this is probably a thing. And then this guy's also from Hawaii, though. So when we go in a couple weeks, we're going to hopefully find a lot more of these guys. And so, yeah. I don't really have a lot of samples to talk very much about this complex, so I'm not going to. But I am going to talk about this guy. So this is the interesting one. This is our local bully here. And they have been found off the coast of Friday Harbor, in Friday Harbor, all the way down here. And what's interesting, there's a lot of interesting things about this network, especially. I mean, you guys are just loving networks, I'm sure. So there's definitely 2 distinct things here, and it's replicated. It's not just one dot way off on a long branch. So what's exciting about this, a couple of things. These guys are really not very diverse, and they were collected from Friday Harbor all the way to the Monterey Bay. So that's a huge species range for these guys to have like basically 4 haplotypes. Whereas these guys are all collected in the Monterey Bay. Granted, it's over a long period of time, but they're a lot, and there's a ton of diversity. Anything else interesting in VC? I'm sure I'm forgetting to say something, but okay. Okay, so what the heck? Why do we have this second thing that is showing up from Friday Harbor to the Monterey Bay? So it has a lot of things. Maybe it's a depth thing. Maybe it's above and below the oxygen minimum zone. Maybe it's an onshore-offshore thing, and we're collecting a bunch of things onshore, and then there's a whole water current other system out there. Nope. So I think the black dots are Friday Harbor, the gray dots are Monterey Bay, and you can see, yep, they're all the same. And we have lots more Monterey Bay guys. And then depth is not a thing either. We find the deep Friday Harbor MITO-TYPE. We find that lots of shallow guys. These are all the blue water dives and dip nets, especially from the aquarium. Thanks, guys. Then when they start to look sad, they give them to us. It's great. So that's not a thing. It's not a depth thing. It's not an oxygen minimum zone thing. And then I thought time. maybe it's a timing thing. These guys live a year. I don't know where they go all winter, Florida or something, Hawaii. So I made this nice little rainbow. So we have samples from 1993. I know you can read that. 1993 all the way till like last month. And I made the shades of gray for the more recent ones. And yeah, there's really no pattern. And the 1993 ones were a bunch of individuals that George Matsumoto collected. But you can see all the colors are everywhere. But our sampling efforts have significantly increased. And being a Moss Landing student, I think about these things. Since I joined the lab in 2017, I was half-time in Steve's lab. And I liked Bolinopsis, so we collected lots of them, and then lots in 2018. And so I blacked out the more recent guys, and still all the colors are everywhere. So it's not a time thing, or at least nothing obvious that's popping out at me. I made this morning, so if anybody sees anything interesting, tell me. So it's not depth, it's not onshore, offshore. I don't think it's time. So we also sequence nuclear genes because we can't just sequence mitochondria, because mitochondria can act weird. They do weird stuff sometimes. And so these are phased H3 data. And so you get your nuclear genes from mom and dad, and so I've pulled them apart and phased them. And so you can see these are labeled by where they were collected. And so the yellow guys were collected in Friday Harbor, and the blue guys were in the Monterey Bay. And so you can see there's certainly a frequency shift. And if I break this gene down into SNPs, there are diagnostic polymorphic SNPs that differentiate between Friday Harbor collected specimens and Monterey Bay collected specimens. And I also have several other nuclear loci, preliminary results from several other nuclear loci that are saying the same thing. And so I do believe that these are distinct species. That's why I crossed out potential. I think that we have agreement between nuclear and mitochondrial genes, even though the nuclear genes are much less polymorphic than the mitochondria. So if I label the H3 by the MITO-TYPE that they have, so if it was a Monterey Bay specimen, but it had the Friday Harbor MITO-TYPE, it gets yellow now. And so you can see that when you phase the two genes that you have some, not just ancestral polymorphisms like in here, but you have some potential hybrids that pop up in there. And so I have a lot more work to do on this. I have a lot more sequencing. These are super preliminary results. And Sanger genes are kind of like our rad, high-waisted mom jeans from the 80s. Like, they're comfy, they keep the muffin in check, but they don't always look that good. So we also do RAD-seq, or shotgun sequencing. And so this is like a super filtered down data set where, but I get the same graph whether I have 10,000 loci or 275 loci. And so these are mostly nuclear data, although I just found out today I might be able to assemble some mitochondrial genomes out of some of these data, which is super exciting. Anyways, so you can see, and so this is a structure file. And so each individual bar is an individual that I sequenced for all its nuclear SNPs, and maybe some mitochondria. And then you give all these SNPs to this nice little program called structure, and it spits out the most probable number of populations that you have. And it's a nice way of looking at really complex data. We also do PCAs. It's kind of like a bar graph PCA. And so what you can see is there are definitely, the most probable answer was 2 populations, which was very satisfying because I had 2 things. And you could see there's a yellow thing and a blue thing and a mixing thing. What was exciting about these data is these yellow things were collected in Friday Harbor, and everybody else here was collected in Monterey Bay. Like, I'm running out of Friday Harbor DNAs. We're going to have to go on a field trip. However, I did half of these guys had the Friday Harbor MITO-TYPEs. The other half of these guys had the Monterey Bay MITO-TYPEs. And so this was super exciting data because this was good evidence that we had cytonuclear disequilibrium. And that just means that the mitochondrial DNA are not agreeing or matching up with the nuclear DNA necessarily. And bonus, maybe this guy, maybe a hybrid. Again, a sample size of 1 sucks. So need more data. And so thanks to Darren, he helped me. We just built some libraries for more individuals to do some shotgun sequencing. We will pull mitochondrial genomes out of that and look and see if there's any rearrangements, because that's another cool thing that ctenophores are super weird about doing, is they love to rearrange their mitochondrial DNA. They're just crazy little animals. And it will help fill in some of these questions too, and look for more of these admixed, potentially admixed guys, along with the Sanger sequences too. I love my mom genes. All right, so yeah, I look at the animals now. Taking lots of pictures of my little balls of goo, and oh, these guys bioluminesce too. I didn't put any video of it, but they're really cool. Take my word. And so now I've learned that ctenophores, this is their mouth up in here. I always thought it was here. This is their not mouth. Let's call it something else if you want. And then they have all these other little interesting bits that are important. And so, yeah, I'm really trying to look at the morphology. It's not my forte for sure, but I'm trying. And so, oh, there's our mouth. They're really cute, and then they use their little auricles, and they stuff their little faces, and they're actually kind of rad little voracious predators. I think we're not supposed to say that, though, huh? That's like saying basil. Oh, geez. Okay, they eat a lot of things. That's surprising that you would think a little ball of goo would eat. All right, so I spend all this time taking pictures. It's so much fun, and what I do is take the picture, and then I freeze it, and then I go sequence it, and then I go back to the pictures, or I'm supposed to go back to the pictures and start looking at them and seeing if I can find any morphological differences. Anybody see anything? So I'm not seeing anything yet, but all these animals were collected in the Monterey Bay, and therefore potentially hybridizing, potentially admixed, probably not the best sample to really start and look. Like when we start on population genetics, I usually start with the two most distant populations and then work in. So right now I'm starting from the middle and looking for differences. Probably not going to see any, but also I could probably look at a cow and a horse and I'd be like, it's the same thing. It's 4 legs. It's fine. Right. Okay, so that is basically what I get to do all day now. I take lots of cool pictures and sequence these guys, and yeah. And so with that, any questions? Well, it's so, I never saw them before I started working in the Haddock lab. Like, and apparently we would see them all the time because they'd like show us on the video, like, no, look, it's right there. But so they are hard to see, but once you start looking for them, and these guys are amazing at finding them. So, but in Hawaii, it's really tricky because the water is so clear. In the Monterey Bay, it's super cool. The blue water diving is amazing. Like you sit there and then like these little animals come at you, and it's like the coolest thing ever. But in Hawaii, which I didn't get to dive our last cruise, and I finally get to dive this time, the water's so clear, you kind of like stare at your diving friends to try to see them and try to pick out critters.
Do you have any clues as to patterns of mitochondrial inheritance in these bugs?
Yeah. So
I mean, is the differentiation indicating mother versus father?
We thought that. There's no indication of double uniparental inheritance, right? Because I worked on Middleist, and so that was like the first thing I saw. I was like, this too? Not again. And so there's no evidence of that. And Bob and I actually had a nice conversation yesterday while I was supposed to be writing my talk about that potentially, like maybe these things are more like plants where they are tetraploid and then they throw out a bunch of genes. And luckily, I had lab meeting right after that conversation, and Darren has sequenced the heck out of these things, and there's no evidence for having any tetraploidy at all, or even, so that's what I was thinking, like maybe it's getting two different, it's cloning, or it's getting two different genomes, and then throwing out the other stuff, and that's why these, but the mitochondria are still these super divergent lineages, but not much. Maybe, but no.
Aquarium hasn't had any luck closing the life cycle or getting them.
Oh no, they totally rear them, and they have them on display. And so these are like these are the ones nobody really cares about, and so I get quite a few of them from the aquarium. They have a different species called LOCALITY genus, and they're amazing. Like, you can't even touch them and they melt. You look at them wrong and they melt, and they have them growing and reproducing. And so we actually collaborate with them quite a bit, and they are very nice and give me their things that look sad.
So I know you were saying that like it's kind of hard to extract DNA from the ctenophores. I was wondering if you figured out if there's like a region, because I know like at least in like some Middle East, like you take their gills, and that's where a lot of like good DNA is to extract from. Is that like, are there any regions of the ctenophore that's good for extracting DNA?
So Lynn taught me this beautiful technique of, okay, you guys know the little transfer pipettes? If you snip off the end of the transfer pipette and then you suck up the teen row is where the most of their little muscles are. But then you also have to be careful, because when I was sequencing H3, I found this amazing like whole group of totally divergent haplotypes, mostly associated with the Friday Harbor species. And I was like, yes, that's it. They're super different. I've never seen H3 this crazy polymorphic and different. Like the Albinaconcha all will be like everybody's one ball for H3. And some of them even share. Like, and those species have been split for around 30 million years. And so 30 to 50. So I was like amazed, mind blown from H3, and it turns out they were amphipods. And so they have parasites too. So you have to be careful.